record. So as I always do, I'd like to review where we're at. So, by week 7, I was envisioning that people have kind of a working grasp of TMB, spatial models, and spatial temporal models. Separable spatial temporal models. By separable, I mean models that can be generated as basically a Kronecker product of the spatial process and the temporal process. And that Kronecker product basically means that the covariance between site 1 and site 2 in time 1 and time 2 is equal to the correlation between those is equal to the correlation between those sites at the same time and those years at the same site. And that's what the Kronecker product is doing.
How many times can you Kronecker product something?
Infinitely. Not literally infinitely. It has a finite dimension for the math you're doing, but for practical purposes, you'll get tired of doing it for working and stuff. So and then this week we're kind of returning, some of the concepts we just touched on in week 4 last. Before I go on, Arnaud Bruce is starting a postdoc here at UW, and you want to say anything about yourself quickly here?
Yes, so hi everyone, I'm Arnaud, so I'm going to work here at UW under the supervision of Jim and Tim Essington. And so part of my work will be to use VAST, so it's perfect that I'm arriving today.
Thanks. This week we're touching on these kind of multivariate spatial temporal models that are, as we talked about on Tuesday, are formed as this Kronecker product of, a spatial process, a temporal process, and some covariance across species. We talked a little bit about the different ways you can generate that multi-species covariance, either as this like trimmed Cholesky, it's sort of the same trick that factor analysis uses, or some dissimilarity in terms of traits or taxonomy, and getting into this sort of modern sort of replacement for hypothesis testing, this sort of variance partitioning approach to multiple hypotheses and sort of a multi-causal world. And then anyway, so some people requested that I present a little bit on this package VAST for vector autoregressive spatial temporal model that I've been developing kind of as I've been working through the content of this class over the last several years. So today, basically, I'll start out with a case study about what, kind of questions I think you can answer with a multivariate spatial temporal model. There's other examples out there, but I'll just do this one. And then I'll do a little bit of overview of the package VAST, and then basically just set us loose with pointing at the single species are multi-species R markdown scripts that I have that are kind of vignettes for showing how to use it. You can look at the user manual. It's kind of a pretty unstructured lab class, but we've got several working examples in the package. And then next week we're I'm getting the last of the simulation codes by tomorrow. So hopefully by Monday or Monday I'll have those some comments back to people about their simulation scripts. And then the presentations, I think everybody signed up. Greg, I signed you up for the last slot. And wait, did you confirm that was okay?
Yeah, that's fine.
I forget. So everybody signed up for our presentation. I'm actually missing the finals week presentation because I'll be at DC for this climate meeting, but Jay will report on. And I think most people have said they don't. A lot of people have said no, they don't want their recordings distributed, and so I'll have to view those recordings, but then I'll just destroy the files. So I'm sorry about. Hopefully, please come to me afterwards if you're not comfortable with that solution. And then the written project. Does anybody remember what I said about when it's due? Has anybody read the syllabus? I haven't read the syllabus in a while. So maybe the assignment is that we all read the syllabus and figure out when the written part is due, but it's due pretty soon.
Oh, it is.
What's that? Yeah, I mean, it's due by the end of the class, which is pretty soon. That's all I mean. Yeah, so and then the final piece, so week 9, I'll be here on the Tuesday, and we're going to cover stream network models, so a different way of defining sort of spatial correlations. And then on the Thursday, we'll cover movement modeling and vector diffusive movement, which is one way of generating sort of non-separable spatial temporal models. And it's sort of a big deal in sort of climate and increasingly in some ecology applications of spatial temporal models. And then week 10, the lecture is basically kind of a review. So I'll just do a quick summary of what I think we've covered, and then I want to open it up basically for course evals, questions, and so on. So that's the plan for the rest of it. Every so often people ask me questions that I tell them, oh, ask during class, and I don't think we often get to those in class because I think maybe people forget about the questions they weren't interested in. So this is probably the most unstructured remaining lab that I'll be here for. So this is also the last chance to kind of do those sort of unstructured follow-up questions that we haven't hit. Any questions before we go on?
Do you want those unstructured follow-up questions now or later?
Yeah, sure. Why don't we start with some of them now? Yeah.
Okay. My question was about sort of like how to implement using nodes instead of the exact locations to simplify the computations. And I was particularly interested in what you do with like spatially referenced covariates in that situation.
Yeah. So why don't we come back to that when we hit some of the vast notation for that. Any other questions that I'm forgetting? Somebody asked about model selection, and I didn't put together any slides about it, so I should figure out where to put it when I teach again. But basically the 2 to 5 minute spiel about model selection is that there's, in my reading, not a lot of agreement or consensus about it, either on the maximum likelihood side or the Bayesian side. And there's a lot of nuances and kind of traps that people can get into. So, there's a laundry list of sort of no-nos, like for instance, doing AIC selection and then with your selected variables looking at a wall test or the ratio of the coefficient divided by the standard deviation and looking at that as a z-score and interpreting a p-value from that. Because of course doing model selection with AIC invalidates any post hoc interpretation of p-scores because you're biasing towards strong p-scores during model selection. So there's sort of a long list of sort of those sort of general no-nos that I assume that people will pick up in other contexts. But specifically in what we've covered here, we've looked at AIC, I think. AIC is a slot in the TMB helper optimized function. And that AIC is technically what's called marginal AIC because it's using the marginal likelihood and the number of fixed effects. In the literature, there's something called conditional AIC that conditions on the uncertainty in your random effects and kind of has essentially it counts the number of degrees of freedom not as the number of fixed effects, but as sort of the fixed effects plus the uncertainty in your random effects from basically well, I won't go to that. But so the claim is that marginal AIC, which is what the AIC that you get naively out of TMB, is optimal for making some decision about predictive parsimony for data within the set of random effects that you're estimating well. And if you want to predict to a new random effect, it's especially important to use marginal AIC, or conditional AIC, which is not implemented in TMB. So, the too long don't care summary of that is that the AIC you get out of TMB is a version of AIC, and there's some belief that it's not the best type of AIC for some of what we try to do. So you should be cautious about naively interpreting AIC using these models. And then there's sort of a literature of Gelman's about, how AIC is an approximation to predictive performance. So if you want to measure predictive performance, just doing a cross-validation experiment is probably the best way to do that. So, okay, so AIC and cross-validation are kind of the main tools we use for model selection. Did anybody have any different opinion about model selection? That's not.
Well, what about, I'm thinking of in terms of like, say you have like, you want to evaluate multiple covariance structures, multiple model structures. Like, you just throw the spaghetti at the wall and then do all subsets of model kind of formulations.
Yeah. Yeah, I mean, I guess my opinion is that, any kind of ecological question that's framed as sort of a model selection question is probably framed in the wrong way. Like, I'm not sure, again, like, I think in reality, there's infinite number of processes, and our models are never correctly specified, and so an either or decision about, inference based on model structure, I think is a recipe for bad inference. But if you're trying to, select a single model to interpret, do an AIC seems like a reasonable thing to do. And then whether you do like full factorial test of every possible model configuration, or whether you do some kind of like greedier selection algorithm where you use like a walled test to figure out what variables to exclude and follow a particular path through the design, I don't know. It just gets impractical to do full, run every possible factorial design. If you've got 10 decisions, each of which could be on or off, you've got 2 to 10, or what is that? I mean, is that 1,000 or 4,000? I always forget. 4,000. 1,000. Okay. So running 1,000 spatial temporal models through model selection is probably infeasible. So, some trick to kind of guide your descent through that, path through that decision is probably necessary.
Could you say something about model fit while we're on this subject?
Yeah, what do you mean?
Like the best way to look at that, is that just something like also like a cross-validation kind of approach, or?
Yeah, like good, you mean like general goodness of fit?
Yeah.
Yeah, Casper has some interesting work about goodness of fit testing, and I think his general claim is that you should simulate, fit the model, simulate data from the model, and then see if, the data exceeds some, is more unlikely by some measure than the data you simulated from the model. And that's sort of a general approach to, kind of like an omnibus kind of goodness of fit for your model. But I've never really gone down the rabbit hole. I've never been forced by reviewers to dig into that too deeply. Instead, I usually just use like Pearson residuals or kind of measures of QQ plots, and we'll look at some of those in a bit to argue that the models are fitting. There's no evidence that the models are fitting horribly. So I think that there's probably more rigorous ways to do it than I'm using. Any other? So we can come back to more questions. And I like I said, I think there's probably a diversity of opinions on model selection, so feel free to jump in. Okay, so as I said, the first one was just kind of looking at a specific example of a multivariate spatial temporal model that I did just because I had it written up easier. So Louis Barnett and I were interested in this question about how we have these surveys of rockfishes using bottom trawls, and of course, if you maybe it's not of course, but if you've got a boat with a cable going about 100 meters down in the water, attached to a, essentially a net bucket, and you hit a rock with your net, you're either gonna destroy your net or destroy something else about your boat. So it's really dangerous to trawl in rocky habitat. There's all these modifications to the behavior of the trawl, like putting rock skippers, which are now illegal. But the bottom line is people don't want to fish a bottom trawl in rocks, and if they do, it damages the rocks. And we've got sonar to detect where there are rocks, so people avoid them. And so it's kind of silly surveying density of rockfish when you can't actually get into the rocks. So there's this kind of question about what's the impact of untrawlable habitat, rocky habitat, on our estimates of rockfish density. And one thing we thought of was trying to do this, like, what if you capture corals and sponges, so like structure forming invertebrates, we'll call them, in the net? Is that a proxy for being close to rocks and then therefore predictive of changes in rockfish density? So can you, and again, like in other fields, I think that, you might similarly have proxies, so like if you're looking for incidence of Zika, like looking at, other, mosquito vector borne diseases that use the same mosquito vector would, seem to be like a reasonable proxy, even though we're not sampling Zika well. So I think this is kind of comparable to other things. So we obtained data for 8 rockfishes, and then the structure forming invertebrates for biogenic habitat. We fit a multi-species model, we interpret the covariance, and we compare single species models with the multi-species. And so if we fit single species spatiotemporal models, and this is just showing, instead of having a big panel with years, multiple years, this is just averaging across the years, just for simplicity of presentation. And structure forming invertebrates is the leftmost panel, and then we've got long spine and short spine, thorny head, you've got dark blotched Pacific Ocean perch and shark fin, and then split nose, striped tail, and green striped rockfishes. And so, this is Oregon, Washington, California, and red's high density, blue's low density. For people that know the system, sharp spine, thorny head, the third to the left is kind of more inshore than long spine, and you can kind of squint and see that, but they're both basically broadly distributed within their habitat. The kind of northern rockfishes, dark blotched POP and shark fin, all kind of look northern, and then the kind of, these kind of oddballs that I don't know that much about, kind of have different densities as well. But the point is that if you fit a single species spatiotemporal model, you get this basically like fuzzed distribution where, for instance, POP, well, they're dark blotched, it basically just looks like it's high density, kind of uniformly high density, kind of all across Oregon and Washington, which isn't, in reality what we think about the species. But there's essentially too little information for the species to pick up on areas with high or low density. So if we fit the multi-species model using that factor model and do we've got 9 species, and so we have the number of factors could be 0 if we want to exclude it, or 1, 2, 3, all the way through 9 factors, where 9 factors would represent 9 columns of the Cholesky, which is the full Cholesky, so it's a full rank covariance matrix. And in this case, we're using that full rank covariance matrix, and we're doing a delta model. So we've got a covariance for encounter probability and a separate covariance for positive catch rate. And we're putting in a spatiotemporal process. So it's the Kronecker product of space, time, and time being independent, independent deviations, and then the full rank covariance among species. And so we estimate a covariance matrix for both parts of the delta model. We convert it to a correlation matrix, and that's what we're showing here. And so the correlation matrix for encounter probability shows that the structure forming invertebrates, long spine and short spine, all kind of positively covary, and if they're negatively covariant with the other species. So it kind of, if you think about this as ordination, there's sort of a block of these Sebastolobus species and structure forming invertebrates. that sort out, and then if you look at the correlation for positive catch rates, the dark blotch POP and sharpshin, the northern species all kind of have this block that's highly correlated. So what they claim is that the dark blotch POP and sharpshin sort out in terms of areas with high density when you encounter them. So you can sort of see already from these correlation matrices that there's sort of, kind of three groups that, are coordinated, that you can kind of sort out from the correlation. One of them is the Subastelotes and SFI, one of them is the northern rockfishes, and then this kind of final group is kind of weakly correlated, and it's just the kind of coastwide rockfishes. Where green spotted is least correlated with the other ones in its group, and that makes sense because the single species model is just showing it having highest density in this sort of California bight. So if you fit the multi-species model, you get a pretty different picture of densities for each species, where essentially, structure forming invertebrates, long spine and short spine, that had a lot of information in the single species model. These three models were like fairly well informed in single species models, and so doing a multi-species doesn't affect results that much. But dark blotched POP and sharpshin are actually really different in the multi-species model, where it's basically saying that they have really low density inshore, which matches our kind of biological assumptions about the species. And it's able to pick up this sort of hot spot that's up in near the Canada border that we see in dark blotched a lot. And so the multi-species model is able to hone in on areas of high and low density much more precisely. And then the split nose, striped tail, and green spotted are all shrunk towards each other a little bit too, like this high density patch in. You bet you remember what that's called. I don't know.
California?
Yeah, I mean, that's about my level too. It's the Pacific Ocean. Yeah, so the multi-species model is picking up on fine scale habitat features a lot more by sharing information within those ordinated groups. If you look at cross-validation or AIC, the delta AIC for the multi-species model is like almost 6,000 lower. So in terms of, AIC, if it's above 10, it's sort of a big difference. So 6,000 is sort of like a, ridiculously big difference. Especially given that it has 112 additional parameters that it's fitting. So it's heavily penalized, but those 112 covariance parameters matter a lot in terms of improvements in model fit. Also doing the 10-fold cross-validation, we can get the log, let's see, so this is the negative log likelihood for each model. So, we had a homework where people kind of made this mistake, a lot of people made this mistake where they wanted to look at which model was doing better, but they forgot that the, TMB is minimizing the negative log likelihood. So as a lower objective function, it's fitting better. So in this case, the negative log likelihood is about 400 units better for the multi-species on average. And given the size of the data set, that translates to like the 6%, 5 or 6% improvement in probability, predictive probability for the multi-species model for predictive. And so, we kind of come away thinking that it's improving predictive performance a lot doing the multi-species model. But if we look at the indices of abundance that come out, so we take these models and project density to the whole coast and sum across that and calculate the standard error of that using the delta method. The single species is in gray and the multi-species is in red. And in general, the trends are very similar. It kind of shrinks. It shares information within groups a little bit. So like green striped and split nose both have a lower value in the first year, and then it goes up and then down again. And so the multi-species model kind of shrinks, basically puts that pattern in the striped tail as well, where the multi-species model it's, the single species is just going down for striped tail in the first years, but the multi-species thinks it goes up a little bit and then down again, because it's, sharing information within that group. But in general, the trends are by eye pretty similar. For the purposes of the models we use these for, it wouldn't make any difference. And disappointingly, the standard error is not any smaller. So, strangely, we're getting better predictive performance, but it's not actually seeming to improve precision for total code-wide abundance, which is sort of a paradox that I don't really. I think what it's doing is the multi-species model is having lower, like a faster decorrelation. It's picking up on finer scale habitat features. And so it's predicting those more precisely, but it's also predicting kind of more degrees of freedom spatially. And so it, the variance, the decrease in variance in the precision is offset by an increase in variance in this kind of fine scale predictions. I don't think that's the way it's supposed to work, but it's just the way it worked out in this data set with the models we're trying out. And then more worryingly, for my purposes, it has kind of a big effect on the center of gravity. So there's this, like we've mentioned, there's this sort of discussion in ecology about whether species are moving north under climate change. And people measure that using this center of gravity, this like measure of what's the centroid of the population. And so if you do single species models again in gray versus multi-species in red, the multi-species actually really affects the inference about the centroid of the population relative to the single species. So, the multi-species is saying POP is further north and Sharpton is further north than the single species model. I actually wish I'd computed the design base to see which of these I trust more now, but in kind of like, I worry that climate stuff is sort of scientifically, as a government employee, you need to be careful about how you talk about climate, I think, and so I probably wouldn't want to use a complicated model to talk about climate. There's not a big gain to doing so. It's probably better just to stick with very easy to defend inference, right, in some cases, and so, I don't see a big gain in terms of shrinkage in confidence intervals, and it's a little bit disappointing that it affects the inference so much in a way that I don't know which one, I didn't at the time spend time figuring out which one I trusted more. Yeah, so basically my takeaway from this was that it was nice, these multi-species models were nice for understanding covariance between species. And they also were improving predictions of fine scale variation in density, so they'd be better for identifying habitat or, doing adaptive survey optimization, anything that cares about predicting density at fine scales. But they didn't seem to be that useful for index standardization because it was more complex without getting you very much, seemingly. And I'm not clear, I'm not convinced that the benefit is worth the extra trouble explaining it in a kind of climate assessment arena. So, okay, so any questions about that sort of example?
I have another unstructured question.
Yeah.
So you use the example of like joint negative log likelihood as sort of a proxy for predictive validity. So is that something you have to do in addition to cross-validation in AIC, or is that on its own sort of sufficient to say that one model is at least fitting the data better than another model?
Well, okay, I'm sorry. So this is the predictive negative log likelihood. So this is a tenfold cross-validation where I'm fitting to 9/10 of data, and then I'm predicting the remaining held out 1/10.
Okay.
And then, I'm sorry, so I didn't explain that well. So this is the predictive performance in that cross-validation sense for each of the 10 partitions of the data that tenfold cross-validation is data.
So we shouldn't use the numbers of negative log likelihood that come out from like the model fit of TMD.
If you're gonna use like the fitted log likelihood to discriminate between models, I mean the whole point of AIC is that a more complex model can hone in on the existing data better because it has more flexibility. And so, I think everybody's on board with the idea that if you're looking at the negative log likelihood for your fitted data, you need to also account for the degrees of freedom of your model. And that's what AIC and DIC and WAIC are all doing. But if you're doing a cross-validation experiment, you're not fitting the data, so you don't need to, you can kind of ignore that discussion of proper degrees of freedom. There is a separate discussion in cross-validation about what's the proper way of retaining and excluding data. So this cross-validation experiment is just randomly selecting 1/10 of points without any kind of spatial structure. And other people want to do kind of spatially blocked cross-validation where they divide up their data and they exclude like one portion, all of the data in one part of the area, and all of the data in another part of the area. And I think those different cross-validation experiments correspond to different types of inference you want to make. So if you're going to predict new data within the same spatial footprint, I think this cross-validation makes sense. If you want to predict a new part, new spatial area that you don't have any data for, that probably makes more sense to do that spatially stratified cross-validation, or spatially blocked, or whatever you want to call it. Is that? Anything else for now about?
Yeah, well, does precision really matters for stock assessment? I mean, isn't accuracy more important?
Yeah, so it's complicated for a couple of reasons. So one of them is that, so in general, in these sort of two-stage modeling approaches, and people feel free to jump in. These are sort of high enough level questions that there's not a right and wrong answer. So I'd love other people's thoughts on this. But so in two-stage models, there's an unusual bias variance trade-off. So like in public health, there's this guy, I think Sazalai, who's done analytic work about the bias variance trade-off, and it's not necessarily the same bias variance. It's not like you want to minimize error. It's true that sometimes in these two-stage models, you care more about bias or more about imprecision.
Sorry, by two-stage, do you mean the delta model, or do you mean using the output from this index standard in a stock assessment?
That's in this context, that's what I mean. Like these modeling approaches where you have a statistical model fitted to data, you extract a result from that model and then treat it as data in a secondary model. I think public health does that a lot. We do it a lot. It's just sort of a trick to deal with such complicated sampling designs that we haven't historically been able to fit the raw data in an interesting model. I think that, I don't know that literature, the analytic literature well, but I actually think in that case, a lot of our fisheries models are just biased, biased at low sample sizes anyway. So, so from a pure root mean squared error perspective, it's probably actually more important to have more precision. Like the bias variance trade-off might be more skewed towards minimizing total error and accepting bias in that sense. But then also on the human side, when you go to a review panel, we want to simplify the process of reviewing the assessment by making sure the inputs are unbiased. So it's sort of, I think the trade-off depends on sort of what's the review process and what's the bias in your secondary model. And those are kind of high-level things that are hard to decide. And then the other piece is that a lot of times when we fit these assessments, when we fit these in an assessment, we have processed additional model and a specification in the assessment model. And so that's reflected in the fact that we can't fit the indices of abundance, like that predicted abundance might be low for all the first years and then high for all the latter years. And in that case, it'll be outside the standard errors of the index. And so some people add in an additional variance, representing like variance and catchability. And so if you're adding an additional variance in your model, you suddenly don't really care about the estimated imprecision of the index anyway. So those are two ways that this kind of multi-stage process that we do makes it really complicated to interpret what the goal of the first stage is. You have any thoughts before we move on? Or?
No, that perfectly answer my question. It's like human versus statistical.
Yeah, exactly.
For funsies, have you ever tried to fit one of these models at the same time as fitting a stock assessment model and estimate the joint negative likelihood together?
Like of all the single species models jointly?
Yeah,
All the single species assessment models jointly, you mean?
yeah, with the delta model estimating at the same time as the assessment model.
Yeah, nobody's done that I know of. Yeah, so Mark Monder one time did the index standardization within the assessment model, and it worked really badly, and he never tried it again, because the raw data was over dispersed, because he wasn't doing a spatial model. And so it thought that the index was way more informative than it should have, because the raw sample size was really big. So that didn't work. And then I've done some stuff, and I'm sure I'm missing other examples. I've done some stuff where I fit a bunch of stock assessment models using the same recruitment index, like basically the dominant factor of a factor model for recruitment deviations. Fit a bunch of stock assessment models jointly that way. And it took a month on like a multi-core machine to optimize. So I published it and then never did it again. So that's didn't really work either. So but it'd be fun to revisit if you want to talk about it. Like for the Seattle model, I mean, I think like doing like multi-species spatial temporal models would be awesome. But figure out how to do that. Yeah, the only effort I did for that was like the multi-species Gompertz model, doing that as a spatial temporal model, and it worked for that one case, but also I haven't published it again because it was too slow. Any other stuff before we kind of move on to the brief outline of the past? Okay, so my claim is that, like NOAA historically, people haven't described NOAA, the National Oceanic and Atmospheric Administration that I work at, as sort of this like, hydra that since the 70s when it was started has sort of moved into a bunch of, and I think any bureaucracy does this just by incentives of bureaucracies, but, it's moved into a bunch of different venues to get money that's, for legislated tasks. And so, some of the dominant kind of legislative tasks that NOAA has are stock assessment, these like single species fishing, sustainability assessments, habitat assessments, protecting individual habitats, integrated ecosystem assessments, for trying to figure out kind of like if we fish down forage fish, does it hurt the whales? These kind of like holistic ecosystem kind of trade-offs. And then climate assessments increasingly. And because they're evolved independently, they end up not talking with each other that effectively. They have different standards of review, and then they end up squabbling over kind of a limited pot of money. And so the goal would be to try to get them all together doing the same kind of stuff using spatial temporal modeling, because I think spatial temporal modeling could offer insight for all of these different kind of questions. And the benefits of doing that, I think, would be, that if we have a spatial temporal model, we can include kind of fine-scale biological mechanism, we can communicate better, and we get similar standards of review. So fish stats is this sort of splash page. I forget if I showed this already. Does anybody remember seeing? Did I put this slide up already in this class? I don't think so. I talked about it a bit, but yeah. So if you go to fishstats.org, you get this splash page that that shows like the vector autoregressive spatial temporal model, some visualization tools, and some data harmonization tools. So it's sort of like the $7 version of the IHV website that's like so much prettier. and I can see why they get way more money than anybody in fisheries does. Yeah, and so VAST is at its heart just built to be this delta model. So we've got one linear predictor, P1, and a second linear predictor, P2, and in a conventional delta model, we've looked at this, it might look like this. Sorry, this. This supposed to be. So yeah, we've got this logit linked linear predictor for encounter probability, and then a log linked linear predictor for density when you encounter it. And each of those linear predictors has this sort of long form, and this is sort of a simplification across some other features that exist, but this is sort of the medium length version. So if we've got a multivariate spatiotemporal model, we have indexing for C for category, S for space, and T for time. We've got a separate, by default, we have a separate intercept as a fixed effect for category and year. And then we've got a spatial process that's structured as a factor model by default. So omega 1 and omega 2 are the spatial variation for those two components. That varies by space and for one or more factors. So if you've got a single species model, like there's only one category, and if you turn on spatial variation, you can only have one category, one factor in it. But like in that 9 species model, that only had spatiotemporal variation, we were fixing 0 factors for this, 0 factors for this, so it drops out of the model. And then we have the full range for the spatiotemporal processes. We have 9 factors for epsilon 1 and 9 factors for epsilon 2. It represents sort of a dense, the full rank covariance between species for their spatiotemporal process. Does anybody want to, this notation we looked at a little bit already, but does anybody want to do a kind of review of that or have any follow-up questions about it?
What's that final sum?
The final.
Lambda times Q.
Okay, so I'll keep going then. Yeah, totally. So there's.
So that's fine.
Yeah, exactly. So there's the factor model for spatial and spatiotemporal, and then this term eta is what I'm calling a Bessel effect or over dispersion, and so it's indexed by some category V of I, and V of I could have as many levels as the number of observations. In which case, eta 1 would have as many, would be length equal to the number of observations, and it would just represent sort of uncorrelated covariance occurring at the sampling level. So in other papers I've interpreted that as sort of fine scale spatiotemporal covariance. So some covariance that occurs at a finer spatial scale than we're modeling, or because of some fine level operations in the gear. In other papers, we have V sub I as sort of a factor with as many levels as the number of vessels in a sampling design. And it represents sort of covariance, some variation among vessels and their sampling efficiency. Are you kind of winding up for a question?
No.
Okay. And then XTP is spatial variation in habitat. Measured variation that we've measured at every location X and year T for P variables. So it could be like bottom temperature or ocean acidity or some distance from rocks, something that we think affects density of fish. And then Q is kind of the notation. It's a symbol that we use in fisheries for catchability. And catchability is what we call sort of the process of determining how many of the species that are at the location you're sampling you actually observe. So like, if you've got flatfish and a bottom trawl, a lot of times you're sampling and the bottom trawl goes over the flatfish because they're buried in the sand. And so domestic violence, I think that people don't self-report, don't self-report unless you've got a very specific design. So if you're combining, like county level measures of domestic violence with panel data following individuals self-reporting it and to somebody they trust, the catchability of the detectability or whatever you want to call it of one of the surveys might differ from the other survey, which results in kind of systematic bias between them. If China was doing a sampling of fertility of women in the 70s, people might not self-report when they break the one-child policy. And so that'd be another case where you have to deal with detectability when combining data from multiple surveys. So Q is indexed by sample, and so if you had a sample of 1,000 samples of women self-reporting their fertility in China, and then 1,000 samples of some panel design that's actually able to count them exactly, you've got 2,000 samples. The first 1,000 might be this, less reliable data source than the other, and so Q could be a dummy factor, where it's ones for the first 1,000 and 0 for the next 1,000. In which case lambda would represent the detectability, would represent the difference in expected data between those two data sources.
Is that not a fixed effect?
Exactly. So I don't have any notational aids here for which are fixed and which are random.
Okay.
But the conventional way I do this is beta is fixed, L's are fixed because they're kind of a variance. Essentially, they're kind of representing the covariance. Omegas are random, epsilons are random, and those are random because you got a spatial process, and you've got a distribution on that spatial process. This L is fixed again because it's the Lodex matrix, and etas are random because they're kind of an additional process of variance. And then gammas are fixed, X is data, and lambda is fixed, and Q is data. So you have to specify the structure of the catchability matrix and the density habitat, density covariate matrix. Any questions? And then to predict densities, we take the estimated values. I did this slide and then somehow I overwrote it, and so I didn't bother to put the hats in, but you take the estimated values of L and epsilon from your model in gamma and beta, and drop out the portions that are not indexed by space, so the kind of catchability covariates and the Bessel effects. You drop when predicting density, such that you're assuming you're predicting density at some reference value for those. And then you multiply those two components together if it's a conventional Delta model. And then from that density, dropping the C subscript, but you could put it back in, you can get total abundance, you can get average density, center of distribution, or the effective area occupied, like we talked about before. Before we were kind of framing them as function valued, with integrals, whereas now I'm kind of doing the summation across the number of knots and sub S, I'm calling it here. So that's sort of the.
So this is maybe where my question comes in, I guess.
Yeah,
so
okay, exactly,
you are
sorry.
Putting in your data as spatially referenced, and you're using those to estimate all of those spatial effects, but then you're predicting separately at a new set of knot locations, or you're sort of assigning knot locations to your data and
Yeah. So there's a bunch of ways a person can do this if you're writing your own code, if you're thinking about it kind of theoretically. So one of them is that, we want to think about the knots as sort of a predictive process kind of interpretation, so I think that we want to say that there's some unknown density function that's, could be queried at an infinite set of points. And then we want to take an integral across it, and, that's not computationally feasible. So instead, we have a series of knots and, interpolate first order, for like piecewise smoothed if it's inla, or in vast world, it's just piecewise constant. And in that case, you could have, you could have the knots, you could have 100 knots, but then you could have the density covariates put in at a much finer spatial scale and just say the prediction of density in any location is the combination of that. So in that case, you'd have this linear predictor would change such that you're summing, such that the x is at a finer spatial scale than the number of knots passed. And so you'd have to do some bookkeeping to make sure that at any location you figure out what the knot is, get the knot effect at a core scale, and then the covariates at a finer scale. And then you'd use that same kind of core scale for the the spatial variation and the fine scale for the habitat covariates when predicting density, here. So that, and I thought about putting that feature into VAST, but it just would be like kind of a bigger code overhaul, and I've never had proper motivation to do it. And if you're going down that route, then it might make sense to do more of a better way of interpolating between knots as well. Instead of my piecewise constant VAST method, the inla piecewise linear makes a little more sense. Or like, and the package MTCV is doing like thin plate splines, so it interpolates even more reasonably. So that's sort of the theory, the broad scale theory, but in VASTland, I'm kind of assuming that the habitat covariates are defined at the same spatial scale as the knots. And so, there's other people who have thought about this, so like Jane Hewlett, who is well known for like being an early developer of Maxent and Russell-Solney methods, or kind of a proponent of them. Talked about how if you've got, if you're modeling at a coarser scale than the biology is operating at, you could put in, and you've got fine scale samples that you're having to roll up to the scale of your model. You could put in, something like average temperature from your satellite measurements at the scale of your data, but you could also put in like the variance of temperature for your measurements within a given scale. Or you could put in like the minimum temperature. Like plants oftentimes respond to like, if there's a frost that defines their kind of maximum poleward extent, so they don't care about average temperature, they care about what's the minimum temperature that occurs for a 24-hour period or something. So there's, in species distribution models, there's sort of this art to kind of coming up with a biologically relevant way of rolling up fine scale samples to the scale that you're modeling at. And so that could, that's sort of what I tell people to do in VAST, is just come up with a biologically plausible way of taking fine scale samples and aggregating them to the scale of the knots. And again, in VAST, I claim that I'd rather long-term just get people to model a finer and finer, more and more knots, so that the model is at a finer and finer scale, and then asymptotically the whole problem goes away. As they say, asymptotically we're all dead, so I don't mean, I mean like, I don't know, that argument is not that good.
So on top it's logit -1.
Oh yeah, sorry, so, thanks. Yeah. It's the point, as we talked about before, there's sort of a set of variables that we use to predict variation in density, so including these catchability covariates and the Bessel effects that we use to explain the data, but we don't condition on them when predicting. And so it's essentially a way of filtering out some portions of the variance when we're doing predictions. And that filtering process matters if Q and eta are non-random with respect to something, then that filtering or not filtering will, cause a substantial change. In terms of diagnostics that are built into VAST, I kind of have features to show people what's the, that where they're extrapolating to in latitude and longitude and UTM and what the location of knots are, just so people can make sure it makes sense. The distribution of data. So there's sort of a literature about preferential sampling. If the data is only occurring in some areas, in those areas, if you have more data where there's some amount of difference in your density, then that can bias spatial modeling. I also like plotting the data because it shows if you're extrapolating to areas with no data, which is reasonable to do, but makes you have to be thinking more clearly about what your model structure is. And then I have sort of a QQ plot for the positive capture rates. This sort of predicted versus observed encounter frequency for the presence-absence part, with a shaded area representing sort of 95% confidence intervals for that. So Arnaud, has worked with VAST, and this has been sort of sufficient for us to kind of claim that we have checked, model goodness a bit. And then there's also, you can look at Pearson residuals. So a Pearson residual is essentially taking your error and kind of standardizing it, transforming it to like a z-score. So anything above 2 or below -2 is sort of a substantial. And so in this case, this is for the Pearson residuals for encounter probability for some species off the West Coast at a core scale. And there's a lot of dark reds representing cases where the encounter probability is much higher than the model is predicting, well, much meaning somewhat higher. But there's not really any persistent spatial pattern in those residuals across time or in blocks of areas. So I don't think the model is systematically missing in the north or the south or in 2003 or whatever. So in reality, sometimes people take their residuals and plot them, like do a semi-variogram on them, or you can start doing these like parametric bootstraps where you simulate from your fitted model and compare it with the data like we were talking about. And then of course, like just looking at the gradients. So, with the Newton, doing a couple Newton steps after the model fits, we get all the gradients down to like 1/1 billionth, like we kind of talked about. So anytime, I present results for applied audiences, I want to make sure that there's absolutely no risk that it's not converged. So I just spend the extra time really drilling results down to a small gradient. And anyway, just checking that all the MLEs are away from the lower and upper bounds and moved away from the starting values is important. So it's just important to get in the habit of actually looking at the final results by eye. And then in terms of problems that people often have with VAST, probably 90% of the problems people report is because some combination of species and year has a 0% or 100% encounter probability. And when that happens, the intercepts for what I was calling the beta 1 are not defined. I mean, they either go to negative infinity or positive infinity for that year species. So it's just a problem with the delta model, and there's sort of a series of fixes that we've kind of kludged together, but none of them are very satisfactory. And then if parameters start hitting bounds, I mean, just check, checking the parameter estimates, making sure that the geometric anisotropy is not causing a problem. So, this is a lot of stuff that people hopefully have kind of started developing these skills of, if you have a model that doesn't fit, there's a series of sort of steps you do to try to figure out what's the problem and how you change your model. So that's that's all the content I wanted to cover. So like I said, the remainder of the class would be looking at the single species example and the user manual if people are, well, the single species and the multi-species examples and the user manual if people want. So why don't we do people want to work individually or in small groups?
I like working individually.
Yeah, I mean, as modelers, I think people get used to working individually. Okay, and how about, who wants to work on the single species? Raise your hand. A couple and multi-species. Okay, well. So I just hope that we have at least a few people to look at each of them. So what I'm gonna do is so that link takes you, you can go to fishstats.org, like I said, and that takes you to the splash page. You go to VAST, and that's a GitHub page. And the way I've organized it, I have the main page written here, and so you can look through it if you want. But for our purposes, I've just put stuff in the examples folder. So, for people who want to look at the single species, I recommend you download the R Markdown file and just start going through it piece by piece. And it's written to kind of have an explanation of what each block of code is doing. Same with the multi-species. And then maybe we spend a 10-minute break and then people spend 20 minutes kind of working individually, and then we try to report back with the benefit of the single species is that there's multiple data sets that are loaded with the package, whereas the multi-species, I think you have to be, you have to spend a little extra time to kind of get off the prepackaged example. So the multi-species looks a little bit, this is formatted like R Markdown on GitHub, but you can change the set of species for the Eastern Bering Sea by putting in new species names here. So for the people looking at the multi-species, I recommend you just, if you don't know fish, then you probably stick with these example species. But if you know anything about the Eastern Bering Sea, you could, look up a few other of your favorite species, like, there's halibut and Pacific cod, and I guess that's the only two I can think of offhand. No. But, you can come up with some other species to look at and just change it in that species set. So does everybody kind of clear on what the goal is for the next half an hour, 40 minutes? Okay. So yeah, so I'll take a break. I'm gonna walk and get coffee just down the way, and people are welcome to take a break as well. I'm back. Well, so going to the examples folder, and downloaded it onto my own machine. And going to the single species directory and ignoring updates as I always do because I don't have install privileges on my machine. Government. This first block is installing the packages and then loading the packages, and and there's some stuff at the top that's sort of formatting stuff for when you make a PDF that I'm ignoring. There's some description of the software and some list of citations for follow-up stuff. And in this case, the data set vec, this is just a character vector that's listing all the different data sets that I have kind of distributed with the package. Try to have one data set per region I've worked with so that they have something they can go back and look at later. So in this case, the sixth element of this is Eastern Bering Sea pollock. So I'll just run that as the default one. So data set equals UBS pollock. I'm using, for backwards compatible, for better or worse, I kind of force people to define the CPP they want to work with. So it's the 4.1.0 using semantic version numbering for the CPP that the package is using. And all of them are kind of backwards compatible. But I kind of wanted to keep those up there so that if somebody comes back years later, they can use the same CPP that they used originally in case there's formatting changes. So they can plug the old results in directly and get the same, slots back. Then the mesh, you make decisions about the spatial model being used. So mesh, like we talked about, mesh kind of refers to the triangulated mesh. That's kind of the keyword. So like the SPE method with NX being the number of SPE station knots, essentially. And then this other stuff, grid size, and it doesn't matter. The k-means is how the knots are being decided, and that's something I'm leaving as the default. And then for a single species model, field config, using the notation that we saw, like omega being spatial, epsilon being spatiotemporal, and then the 1 being the presence absence, or the first linear predictor, and 2 being the positive catch rate linear predictor. 1 is like R code for true. So it's just saying, yeah, leave in a spatial and spatiotemporal process for both components. And then this is just leaving row config at default. Those are like ways of inducing some temporal correlation in different processes, and I won't turn anything on. The eta is over dispersion, and I won't turn any like Bessel effects on. And the OBS model 2.0 is the default. And I won't change that, and that represents like a log normal or a gamma, and whether it's a conventional delta model or a Poisson-Lens delta model, it's, and this is all documented in the data fund help file. So like if you do question mark this function later, you can get some details about what these different things mean. And then the outputs are derived quantities, and so it's, maybe I should get rid of some of these slots. You don't need, if this was an empty vector, it would just turn everything off, but they're kind of, like in this case, I'm telling it I want to calculate shifts in distribution and shifts in area occupied, and just spend some extra computation time computing those. This big block of code is just telling it what, no, this isn't so bad yet, but it's, you could stratify the results. So you could calculate the density field and then also calculate, like on the West Coast, for instance, we might want to fit a model to density for all species, but then calculate an index of abundance for Oregon, for Washington, for California, or for the whole coast. So you can kind of post-stratify your results to calculate derived quantities that conform to a specific sub-area. And this, of course, would make sense in public health, like for global verge of disease, to model all of Africa, but then calculate density in Kenya versus Ethiopia or whatever. Different strata limits for different data sets by default. And then the syntax is set up where it tries to associate each data set with a region. So it's saying, for instance, if I'm using Eastern Bering Sea pollock, I want it to be in the Eastern Bering Sea region, and that's used later. I make a data frame or a directory to save results, and I record a record of all the decisions I made. And this block of code is loading data. So for the Eastern Bering Sea example, down here it loads data that is packaged in spatial delta glim for Eastern Bering Sea pollock. I think we've used the data set before and loaded it this way. And then I'm formatting, I'm just renaming columns basically. And kicking out any rows with NAs because some regions just send me bad data sets. And then the meat of, the kind of harder stuff to kind of package are the different decisions somebody might want to make about the area they want to extrapolate results to. So for some regions with a well-defined survey, I've worked with people to encode what's the footprint of the sampling design. So, like Arnaud and I did this for defining the Gulf of Mexico for extrapolating, or I guess we did this for the Atlantic Ocean as well. But I don't think I ever put it in the package, and we can come back to that. But for the Eastern Bering Sea, there's this prepare extrapolation data fund, looks for different predefined regions, and basically makes a list with a bunch of random shit in it that represents the area that you'd extrapolate your results to.
Would that be like creatable without that function? Presumably that function only works with like the spatial delta GLMM.
Yeah, exactly. So if you were going to kind of rebuild something like this, I'm sure there's other ways you could factor the code kind of for a given. And if I knew that I would still be using this project, I probably, like 5 years later, I'd still probably maybe would have built the code a little differently in terms of how I broke up tasks into functions. But yeah, I mean that, if region equals other, I guess in the syntax of this model, that means that it's a region that I don't have a footprint that's packaged with the model. So like the Puget Sound, I don't have a footprint representing the survey. So instead, in that case, you can either put in the observations that you have, in any place you sample that I assume is within the footprint of the sampling. And so you could define your spatial area based on those locations and sort of a buffer around it. And that's what this function is doing. Or there's another function that allows the user to put in a data frame with 3 columns, like latitude, longitude, and area. It would just represent a grid. So like if you're doing Africa, you could put in, like global burden of disease wants to do it like a 1 kilometer by 1 kilometer grid or something. So you could put in a data frame with the latitude, longitude, and 1 kilometer squared, representing every grid cell that you want to project to, and just put that into a function here. Okay. I don't have an example of that built with FAST. I mean, if you want to talk about it, we can, you can put it on the issue tracker. Somebody's actually asked it on the issue tracker already, so you could look on the FAST issue tracker and find where somebody asked how to do that. And then, so that's sort of one of the 2 kind of messier pieces of code. I mean, it's not messy on the user side, but it's not really probably optimally factored. Tasks for these 2 different functions could have been built into, more or fewer function calls in a way that would have been more interpretable. But anyway, so this spatial information function is taking that extrapolation list and some other stuff, and it's basically generating what is it doing? It's running the k-means algorithm, and it's deciding on the location, using that to decide on 1,000 locations in UTM that, that's the location of the non-cell model. And it's doing some other stuff, like if I decided to do a grid instead of a mesh, like a 2D AR1 process for space, it would have done something different for doing that. Then I've got this data fund that builds all of the objects together in the list format that TMB expects. And then build TMB fund is a wrapper that builds the tag list of parameters, decides what's random, builds the object, and does some other stuff, kind of preparatory stuff. So in this case, right now, it's compiling the CPP file to a DLL like we have to do for TMB. And on a Linux machine, you might get a bunch of random error messages at this point. I mean, not error messages, what Linux thinks are informative messages, warning messages about syntax that I haven't tried to fix. How far did people get on their own, or did anybody actually get the whole way through one of the scripts?
It is optimizing still.
Okay.
Yeah, my model's running.
Okay. And you guys were within the single species?
I was doing the same, yeah.
Yeah, with 1,000 knots, it's a lot slower. So maybe for purposes of demonstration, I could have suggested that people do fewer knots or something. Yeah, so, the TMB list outputs a bunch of stuff, one of which is the TMB object. And the other stuff is like some of the intermediate things that function calculated. But, we can just do what we're used to doing. And running obfun and obfpar to run the first, to convergence to inner optimum to get the Laplace, approximation of the negative log likelihood. Got this little table that it outputs that lists the number of parameters. So if it's using the same notation we used before, so you can kind of check that the model is structured about the way you expected. And in this case, we're estimating 33,000 spatial temporal random effects. So it's almost 100,000. Well, I guess it's almost 70,000 random effects. So of course it takes a little while. We didn't really talk about it, but the bias correction, we talked about bias correction a little bit, and can anybody remind us what the bias correction is doing?
For any nonlinear parameter transformations, it's doing the delta method to
Yeah,
an unbiased estimator.
yeah, it's sort of essentially doing something like accounting for the bias using sort of a Taylor series expansion. It's a little bit more precise than a Taylor series expansion for the bias correction of that nonlinear function.
That's just for the random effects?
So it's any nonlinear transformation that includes random and fixed effects will be biased, yeah. In an MLE, typically, you also get bias for a nonlinear transformation of your fixed effects, but the kind of claim here is that we don't typically bias correct the fixed effects. I don't know, that's just not something that people are in the habit of doing, but I wanted to make sure that the random effects are bias corrected. Under asymptotically, your fixed effects are consistent and go to the true value, and so if the standard error of your fixed effects is really small, then the effect of bias correction is infinitely negligible. So it doesn't matter for consistency, but low sample sizes, there's sort of an argument for bias correcting both the fixed and the random effects. But anyway, so here I'm turning on bias correction. It's really memory intensive for some functions because it breaks the sparseness of the random effects sometimes. And so in this case, we've got this new feature where you can just bias correct some subset of the derived quantities. So in this case, I'm just bias correcting the index. I'm not bias correcting everything else. And so that makes the bias correction run a little bit faster. Or, well, it makes it run with less memory. So I'll just start that running. And then there's this big block of code that runs individual diagnostic plots, that we looked at. So a QQ function plots the data, plots the Pearson residuals, gives you the AIC, plots the geometric anisotropy, the density, and so forth. So all the plots should be pretty straightforward. So anybody want to, Simon, I think you were the one who, expressed an interest in walking through it. Was there a particular part that you want to come back to? So.
No, I think the lists of the input data were really big, and I And like, since the creation of them was all wrapped into functions, I was kind of like, oh, how did we get here?
Totally.
Is it just like something, I don't know, like I would like to
Yeah.
figure out, but I think it would probably take some time on my own to look through each piece and
Yeah.
actually gets put into the model.
Yeah, I mean, that's the problem. I mean, I don't think that this is horribly designed, but I think that the way I factored the functions, it's not really clear to people intuitively what they go in and change. You know, so like the benefit of the R structure, having everything in a single function with few inputs, is you're like, okay, well, I can just change the formula or I could change the data. And this longer script has a lot of different places, and if you change, you know, if you change something in the wrong place, maybe it's already been used, creates some other object, and then you just get some error in your function. And so I think for sharing software, this doesn't conform to sort of best practices for.
No, yeah, I mean, I think for like showing example, it's fine, but as far as like thinking like, oh, I want to replicate this, but not necessarily use the pre-made functions to prep the data.
Totally.
And that's just something that's on me to spend some time to figure out.
Yeah, well, yeah, totally. And then in terms of the executable, it's, last I remember, it's about 1,000 lines, which is obviously a lot. But let's see. About 400 of it is doing different outputs, and a lot of that's sort of like multi-species measures of synchrony that I haven't talked about and haven't documented. It's in review. So, like Grant, I think you're, like with the Seattle model, you know, I think that this is big enough that at some point I could start breaking it up into different scripts, you know, or functions, and sort of, it'd be interesting to talk about best practices for how to make it more readable that way. But, you know, so maybe let's say a third of it's output stuff, and you know, about a third of it is, you know, basic functions at the top, and then reading in, reading out. So you know, there's maybe 300 lines of kind of working code, and I'm not going to go through it piece by piece, but you know, really, you know, people should be able to read, you know, 95% of this, you know, using the stuff we've covered. So like, you know, I define a precision matrix Q1 and Q2 for the two different delta model components. I define I do this QSPDE function to convert a tag list SPDE that just has the 3 different matrices for that SPDE method. And I save it to this Q1 or Q2. So that's sort of an encapsulated function that's, I think, up at the top of the file, if I remember right. I then convert that to a Gaussian Markov random field and save that. So that's sort of a technique that we haven't seen before. And I pass that GMRF to a function to calculate the negative log likelihood of different Kronecker products of categories, space or space and time. So there's a little bit of more like function, you know, embedding what we've done before in functions. So I don't have to write it out, you know, when I do it for both components of the model. But for the most part, I think that it should be, in broad strokes, readable.
Compiling the model fails for me.
Well, I'd be happy to look at that with you. I'm not sure why that would be.
Are you doing multi-species or single species? Multi. Yeah, my R crashes on data fun.
Well, I'll have to look at it, yeah.
It doesn't give me anything.
Totally, yeah, Windows, so that's some overwriting problem, and I haven't looked at the multi-species for a while, so I'll have to look at it. Well, anyway, it's not that interesting to watch it converge, and it's taken a while, but,
Can I ask you some random CMB questions?
yeah, sure, of course.
Just thinking about the difference between using something as like a data array versus a data matrix is the only benefit that with a matrix you get to do matrix operations.
Yeah, essentially, and the drawback, of course, is that it's restricted to 1 or 2 dimensions.
Right.
So yeah, sometimes if you want to do matrix multiplication, you can take a slice out of an array and then coerce it to a matrix and then do matrix multiplication on that. But, as we've talked about, modern compilers, you can loop through the matrix multiplication and it won't really lose you any speed unless the matrix is sparse and then it's able to detect sparseness during the matrix multiplication. So, really the main trade-off is what you think is more easy to read.
Yeah, that was my other question. There's no, because it's compiled, there's no benefit to trying to vectorize calculations. You could just loop it.
Yeah, I mean, that's my understanding. It's been a while. I'm sure there's exceptions to that.
Right.
So like I'm saying, like the eigen package, if you do matrix multiplication of 2 sparse matrices, it'll be a lot faster than loop it. I think. But,
I have sort of like a laundry list of questions about optimizers, but I don't know if that would be appropriate now or better for another.
well, I mean, I'm fine talking about it. I mean, we've kind of covered the content that I wanted to. I'm sorry that the multi-species aired out for you guys. Obviously I should have checked that before starting class. But,
I'm gonna try it one more time.
yeah, I mean, it's possible there's a bug. I mean, I just was doing a multivariate model, and it's possible that the latest version had broke. I've got a Travis CI, but that aired out because of some changes I made for New Zealand. So.
I mean, I'd be curious to hear your optimization questions.
Yeah, go for it. Yeah.
Oh, so just sort of, I should open up like the Optimix stage, but just, there's a lot of options that you can specify. So like the ones I'm thinking about are like iter max, eval max. There's ones about like limits in terms of like how close or far you get. And I've seen a lot of things online about just sort of general guidelines about these, but I'm wondering if you have recommendations. Because I think iter and eval are like, have something to do with the number of times the inner optimization versus the outer is performed.
Yeah, well, no, it's okay, so, and you mean, so it's a little complicated because there's an optimizer, like we've been using TMB helper, which internally is using a function called NLM-INB. It's a Nelder-Mead, a gradient-based Nelder-Mead. It was built by Bell Labs, and it's a good one, but it's not well documented. So you can do like a control input that includes eval max, or max it, and well, I forget what they're called, max it, and
the ones I know
no,
are
it's
like
iter max and eval max, yeah.
Exactly.
And then there's also kind of similarly named ones that are built in a TMB. So TMB, you can pass some functions that control how it's automatically doing the inner optimizer. So there's kind of a hidden backdoor where you can actually tell the TMB to do the inner optimizer using any optimizer in R. And it's not that, I've never found a case where it was really necessary to do that. But you can also tell it to make sure the inner optimizer is really tightly converged, which means that the gradient calculations will be better to more decimal places. Or you can tell it to, by default, if it does 10 inner optimizer loops, it doesn't improve the log likelihood more than like, the joint log likelihood more than like 0.0001. After 10 iterations, it just sees if it's positive definite, and if it is, it just calls it converged. You can change that behavior. So there's a sort of a set of behaviors that the inner optimizer, the TMB, is automatically doing that controls that behavior. And then there's a series of controls that you can pass to like NLMB or Optimax or Optim that controls the outer optimizer. And which of those are you interested in?
So I guess my questions are sort of the controls that would go into like NLM, INB, found an example where NLMIND was like not the best optimizer for your problem?
Yeah, so okay, so the you know, NLMIMB is just doing the outer optimizer, so it's just, that's only affecting the outer optimizer, and you know, in general, like itermax and evalmax, for really big nonlinear models, they're both set to 200 by default or something, and so it's definitely the case that I've had models where 200 iterations won't finish it, and so it'll give up kind of looking sort of loosely converged, and it's really annoying that it has, in my opinion, that it has that default behavior, so TMB helper just automatically increases that eval max and intermax a lot for the outer optimizer. And I forget some of the other controls it has. I've never found that useful. It has some controls related to behavior where 0 means something, but most of our likelihood models, the scale is arbitrary, so those don't apply.
And so things like, so I see like absolute tolerance parameter, relative tolerance
It's-
parameter.
Yeah, so exactly. So the abs_tol is sort of what it does as the objective function gets close to 0. So if you've got a loss function where 0 has some absolute meaning, it's useful to have some behavior mapped out there. Like in physics, that might matter, but for a likelihood, it doesn't. Those abs ones don't really mean anything to us. And then the rel_tol, I forget what the default behavior is.
It defaults to 10 to the negative 10.
Yeah, exactly, which is close to like the square root of the machine tolerance, which is sort of a good measure of sort of like a noisy function that's propagated like at machine tolerance. And yeah, that seems like a reasonable, that seems like a plenty good tolerance for our uses. Yeah, and then in terms of NLMINB, there's a function called OptimX that it's actually a package that builds together a bunch of like a long list of nonlinear minimizers. And you know, like I showed a few of those at the beginning in one of those early lectures, like you know, it saves out the path of the optimizer, and I showed little videos in one of the early lectures of a few different ones. And the Nelder-Mead is just, you know, across a large variety of models, the gradient-based Nelder-Mead has just kind of been the most robust, I think, is what a lot of people have observed. You know, the problem with the Nelder-Mead, like any optimizer, is it assumes that all of the dimensions are approximately the same scale, and that's why, you know, taking a log of your parameters oftentimes makes sure that it's kind of the derivative with respect to one unit in any direction is kind of within 100 of one another for all your parameters. The Newton step at the end is sort of a scale-free step. It's using that, you know, it's using the curvature to kind of figure out how far to go, and so it doesn't care about scale. So I think that's one of the reasons that the final Newton step, doing a few of those, it really drills you in much closer, because it sort of does a few steps where the minimizer doesn't have to care about the difference in scale between parameters. Or the correlation between parameters, because it's, you know, it's guaranteed to be quadratic around the, asymptotically it's quadratic around the MLE, and so that quadratic step at the end, the Newton step is a very precise way to improve it. So anyway, so doing a gradient-based Nelder-Mead and then that final Newton step is sort of the most robust thing that I've encountered.
Okay. How about for the inner optimizer for TMB? Are there parameters that you like to set?
I think that in the function I used, I'm trying to remember if I used to play around with this a lot more because I used to do like much hairier nonlinear models, and I've kind of become happy to use kind of close to linear models. So this, in this function, I compile the object and then I overload some of the inner optimizer controls. So like I think that the max for the inner maximization problem by default is like 1,000. And so there's sometimes where it calls a really horrible value for fixed effects, and it'll spend forever trying to do the inner optimum problem, whereas this tells it to give up much faster than early. But it also tells it the rel_tol, I think by default is like 1/1 millionth or 1 e-6, and I tell it I want the relative tolerance to be much tighter than the default for this model because I want the gradients to be, for that final Newton step, I want to make sure that the inner optimum is really tightly converged. I forget what par scale does or report does. I think report's just telling it I want it to do this like verbose output of the inner optimizer. So, long story short, yeah, I play with the inner optimizer a little bit with fast, but I don't think it's usually necessary. Here's where the inner method Newton, the Newton is what they ship with TMB, and here you can just put NLN IMB here, and it would tell it to do the Nelder-Mead to solve the inner optimizer. Or you could come up with your own optimizer and just plug it in here. But I've never seen a problem where that was that helpful. The Newton, and there, we could go on about that, but it's part of the weeds. I mentioned briefly that there's this new feature where you can normalize the Gaussian Markov random field only when doing the outer optimizer. So the idea is that the multivariate normal has a constant of integration that's the determinant of the precision matrix. And that precision matrix is made up only of fixed effects. By definition, the variances are always fixed. And so you would never change anything about that Q matrix during the inner optimizer. And so because those parameters are the same, the determinant is going to be the same. And so you don't actually need to calculate the determinant while doing the inner optimizer. And so there's this kind of neat new feature where you can turn off the determinant calculation of the multivariate of the Gaussian Markov random field when doing the inner optimizer, and only do it when doing the outer optimizer. It just saves computation time during the inner optimizer. So that's sort of a feature that's pushed in here. If you have late versions of VAST, and you've got one of these non-standard options. But so far, I haven't found that to actually speed anything up, so it's not something I recommend. I kind of keep hoping they'll improve that feature, so I leave the code in. But yeah, that would be worth probably checking out, or whoever is in charge of the TMB code for the global version of TMB.
Could you say, I'm sure you said this a million times already, but what the optimizer's spinning out again? Which?
Well, fortunately, the optimizer is still running because it's a really slow problem with the desktop. So under the way that I've set it up with this verbose output, the outer MGC is the outer maximum gradient component. So an outer optimum means you've got a vector of gradients of the fixed effects of the Laplace approximation, the negative log likelihood with respect to fixed effects, and the maximum of that with respect to any of the fixed effects in this run was 0.006. So pretty close to converge, close to MLE. The U step, this inner optimizer does iteration 1, 2, 3. The U step is close to 1, meaning that it's close to quadratic. So the inner optimizer, the Newton inner optimizer, is working efficiently. If you've got a U step that's really far from 1, that's when using a different non-Newton inner optimizer might work better. Although I haven't ever really seen a case where it mattered. This inner MGC is the maximum gradient component of the inner optimizer, and it's stopping, for instance, because I told it I wanted a tolerance of 1 e negative 10. Each of these inner maximum gradient components is less than that. So, 1 trillionth or something. So getting that inner optimizer really tightly converged means that I get a really precise version of the outer gradient for use in the Newton method. Because, errors compound in this nonlinear function.
And the value, sorry, go ahead.
No.
And the value itself is the.
And this value is the joint log likelihood. I mean, it's occurring in the inner optimizer, so you've got kind of a hint that it doesn't know the Laplace evaluation, so that the only value it probably is their joint log likelihood. So this, if you run report and report out that JNLL, it'll be about this value. Whereas the marginal log likelihood is not being listed in this part. And the fact that it's not listing the outer vector chain is your hint that it finished the NLM-INV step and is now doing the Newton step, where it's doing a bunch of gradient evaluation, so we're never going to do one Newton step and drill the answer. I mean, yeah, so I am happy to keep doing these questions. I mean, the fact that this is like stuff that is coming up to people, I think indicates that, we're kind of getting to kind of higher, hopefully people understand the big picture concepts and we're getting into these sort of second order questions that actually deal with details of how it's computationally working. I'll interpret it as a good sign.
G.
Yeah.
I think it's a question that will interest Grant because we talked about that last week. Is it easy in TME to use predefined functions to code smoothers like you would have in MJCV? You have like already functions that allows you to do that?
Totally. Yeah, so I don't know. So it depends on what you mean, of course. So on the shipping with the TME, like TME has a bunch of C++ code. And like ADMB used to have code where you put in like that you want a smoother and you put in vector parameters and it would just generate that, it would do all the computation for you. I don't think that TMB has the C++ code encapsulated to do those smoothers. But you can still do those smoothers in TMB, it's just you have to build more of that function in the CPP code on your own. And then of course, did you explore that? Did you explore this with your stuff?
No,
I forget.
I think it's Laura.
Yeah, Laura, yeah. And so Laura's done it, and other people have done this too, where you run MJCV like the spline function, and you extract the basis, this basically a matrix that represents the basis of that spline. And then you put that matrix into like XXTP, you put it in as a covariate of fixed effects.
So that's probably the easiest way to go, right? Rather than trying to find the C++ code that you would have to write in CPP file.
Yeah, of course it depends on what people are comfortable with.
Yeah.
But yeah, I think for a lot of users, that's the easiest way to do it, is generate this spline, matrix, the matrix representing spline using some external package and just pass it in as a covariate matrix. And we've had several different scripts that have had a covariate matrix where we typically just have an intercept or an intercept plus log depth or whatever, but that could be an intercept plus a design matrix for the spline. So in theory, people could work that out, but I'm not showing any details of how to extract the spline basis from the MJCV. That would be something people have to do, work out on their own, or email me and I'll just refer you to Laura, probably.
Yeah, but that's a neat turnaround to
Yeah,
export the
totally.
results of MJCV rather than.
Yeah, exactly. Yeah, so MJCV does a lot of this stuff very quickly, and it does it by defining splines using a Q function, or like essentially a precision matrix. And the precision matrix is oftentimes called Q because it's sort of the quadratic form, like the Q goes in this sandwich matrix for computing a penalty. And so you can do most splines by exporting something like a Q matrix that sort of looks like a precision matrix. And that's kind of on deep level sort of why a spline is kind of like a Gaussian random field. They're both kind of I agree that's interesting, and it'd be fun to build more features like that into Fast.
If there's no other questions, I could ask about the question on my homework, which was how to add the likelihood from the initial state when you have when you've done this is Jay's comment on my homework. Do you remember? It was like,
Yeah.
to a point off, I think, because I had I was adding to the joint negative log likelihood, like the density of the initial state, and you're saying that, oh, that's not what usually what people do, but I think you used it in your code.
Yeah, I mean, the point is that one of the things I've tried to demonstrate is that you can define a model recursively, whether it's recursively across time or whether it's recursively across space. And we'll do this again with the stream network, where literally you're sweeping upstream to downstream. You can define a model recursively, or you can come up with sort of what's the joint probability of that model. And if you define a model recursively, it's really clear that you've got like t plus 1 given time t, but it's really clear that you have to define what it is in time t. And that's, people call that like initializing the model. And if you've got a fixed effect representing the initial condition, then the first random effect in that time doesn't really mean anything because the fixed effect will snap up all the variance for it. So if you defining something recursively in time, oftentimes people will define the initial condition as a fixed effect, and then just the time 2 given time 1, time 3 given time 2, so they won't have any random effect for that same year because they have a fixed effect. But it's hard to do that when you define it jointly, because the joint parameterization kind of automatically includes all years, including that year that you got a fixed effect for. So that's why I think both exist in the literature, because when you do it jointly, it's hard, and put in a fixed effect for the initial condition, it's hard to, it's non-intuitive how to reduce the, is this being helpful when we talk about it this way?
When you say jointly, do you mean like using, like explicitly filling out a covariance matrix?
Exactly, yeah, like evaluating the probability of all of the random effects simultaneously via their joint covariance matrix or joint precision matrix.
Okay, yeah, because so what I had was the recursive model.
Yeah.
But adding, and I had like a fixed effect for the initial state, but also, maybe I just coded it up wrong or.
Well, no, I think what you're doing would be equivalent to having the joint probability of a random effect in every year plus a fixed effect for the first year.
Yeah.
And because you're defining it recursively, it's really easy to see that you can strip out that first random effect and just define the random effect for year 2 given year 1, year 3 given year 2. And it would end up with a little bit easier to interpret the standard error of the first year. The problem with defining it jointly with a random effect in the first year and a fixed effect in the first year is that you've got a sum of 2 different things. And so to calculate the standard error on that sum, you have to do this.
Okay.
A sum, that you have to do the delta method on the 2 parameters. Whereas it's easier if you just have a fixed effect for the first year and then a random for the second, given the first. It's easier in a model to extract the standard error of that fixed effect and treat it as the standard error of your state in that year.
Okay, that makes sense now.
I actually think both will get you to kind of the same thing. So my email response is just like a little bit of meh. But I'm sure there's a statistician out there who cares a great deal about it.
That's not a hard thing to find. Statistician who cares about
Yeah.
anything.
Well, I'd be fine calling it a day. I'm Melissa to stick around and keep answering questions. Okay. Thanks, everyone.
Thanks.